Did you ever think about what's happening inside every single cell in your body? You'd be amazed. Each one has its own unique set of instructions written in the language of RNA. And today we're going to decipher it with single-cell RNA sequencing, or single-cell RNA-seq. Hey guys, welcome back to Bytesized Bioinformatics, where we dive into the fascinating world of bioinformatics. I'm Oscar, and today we're zooming into the microscopic world of our cells with single-cell RNA sequencing. So traditionally, biology often treats a tissue sample like a smoothie. You blend up a whole bunch of cells together and you analyze the average result. This is what we call bulk RNA sequencing. The problem? Well, just like a smoothie, you lose the individual flavors of each fruit, or in our case, the unique properties of each cell. All the rich detail about what makes each cell unique gets lost in the blend. This is where single-cell RNA-seq comes in. It's a game changer as it lets us study the RNA of individual cells. Here's how it works. The journey starts with a tissue sample. We need to isolate single cells from this tissue, which is a bit like trying to find individual characters in a crowded scene. To achieve this, we use techniques like fluorescence-activated cell sorting (FACS), which can sort cells based on specific characteristics, or microfluidic devices that can trap individual cells in tiny droplets of liquid. Either way, the aim is to get our tissue from a bustling crowd into a lineup of individual cells. Next, we need to access the cell's transcriptome, a set of all RNA molecules or transcripts in one cell. This is the cell's working instructions. To read these instructions, we convert the RNA into a more stable molecule, cDNA, through a process called reverse transcription. Because we have such a tiny amount of material from each cell, we then amplify this cDNA, which is like making lots of copies of the instructions so we can read them more easily. Now we're ready to sequence, to read these instructions letter by letter using next generation sequencing technology. Finally, we've got a massive amount of data from sequencing. To make sense of this, we need to analyze the data. This involves figuring out what genes are being expressed in each cell, categorizing cells based on their patterns of gene expression, and understanding what these patterns tell us about the cell's function. We can even trace the lineage of a cell, figuring out what type of cell it came from and what type of cell it might turn into. This is like piecing together the cell's past and predicting its future. Single-cell RNA sequencing is revolutionizing how we understand biology. It's providing an in-depth look at the cellular world, allowing us to appreciate the complexity and diversity of cells in different tissues. With single-cell RNA-seq, we can discover new cell types, understand cellular development in response to diseases, and much more. Whether it's cancer research, neurobiology, immunology, or developmental biology, single-cell RNA-seq is making waves and opening up new possibilities for scientific exploration. And that's a wrap on single-cell RNA sequencing. Thank you for watching this episode of Bytesized Bioinformatics. If you found this video helpful, leave a thumbs up and don't forget to subscribe. I'll see you in the next episode.